everyone, welcome to our YouTube channel, The Exploring Medico. So today we will discuss about the second part of plasma proteins. So in this session we'll cover up the functions of plasma proteins, synthesis and the factors affecting it. We all know plasma proteins are of three types: albumin, globulin and fibrinogen. Now we'll discuss about the functions of plasma proteins one by one. First one is exert osmotic pressure. The protein molecules are unable to pass across the capillary membrane and consequently exert colloid osmotic pressure of about 25 mmHg on capillary membrane. As we all know, the thickness of capillary is very less, so only about 1 RBC can pass in line. So protein molecules are larger in size, so they are not able to pass from the capillary membrane. It is very difficult for them to pass through the capillary membrane, so they exert colloid osmotic pressure of about 25 mmHg. About 70 to 80% of the osmotic pressure is contributed by the albumin fraction. As we all know, albumin are more in number, so it is logically proven that 70 to 80% of the osmotic pressure is contributed by them. The colloid osmotic pressure plays an important role in exchange of water between the tissue and fluid. So this osmotic pressure plays an important role from changing the water between the blood and tissue fluid. Then second one is contribution to blood viscosity. Fibrinogen and globulins are significant contributors to blood viscosity because of their asymmetrical shape. As we all know that fibrinogen and globulins are somewhat larger in size, so they are contributors to blood viscosity, and also they have the asymmetrical shape. Their shape is not symmetrical. The blood viscosity plays an important role in the maintenance of blood pressure by providing resistance to flow of blood in blood vessels. Viscosity only means that resistance to flow. So the blood viscosity plays an important role in the maintenance of blood pressure. So it resists the flow of blood in the blood vessels, and due to which blood pressure is maintained, and it is done by the blood viscosity. And fibrinogen and globulins are responsible for this blood viscosity. Then role in coagulation of blood. The fibrinogen, prothrombin and other coagulation proteins present in the plasma play an important role in the coagulation of the blood. We all know that fibrinogen is the main character in coagulation of blood. So the fibrinogen, prothrombin and other coagulative proteins that are present in the plasma, and their main function is in the coagulation of blood. Then fourth one is role in defense mechanism of the body. The gamma globulins are antibodies which play an important role in the immune system, meant for defense of the body against the microorganisms. So they are saying that gamma globulins are antibodies, and they play an important role in the immune system, and this immune system is mainly meant for the defense of the body against the microorganisms. Then role in maintaining acid-base balance of the body. So plasma proteins, as we have discussed earlier, that they act as buffers and main role in buffering system. They balance the pH. Plasma proteins act as buffers and contribute for about 15% of the buffering capacity of the blood. So 15% of the buffering capacity of blood is dependent on plasma proteins. Because of their amphoteric nature, plasma proteins can contribute with acids and bases as explained below. So plasma protein combines with acids and bases. So in acidic pH, the NH2 group of proteins, as we have discussed that plasma proteins have two types of groups, NH2 and COOH. So in acidic pH, NH2 group of protein acts as base and accepts proton and is converted to NH4. In alkaline pH, the COOH group of the proteins acts as acid and can donate a proton and thus become COO minus. So in acidic pH, protein accepts a proton and becomes NH4 from NH2, and in alkaline pH, the COOH group of proteins acts as acid and it donates a proton, so it becomes COO minus from COOH. Okay. At normal pH of blood, protein acts as acids and combine with cations, mainly sodium. So, in the normal conditions, normal pH of blood, protein acts as acids. Then the sixth function is transport function. Plasma proteins combine easily with many substances and play an essential role in the transport as explained below. So the various substances and various gases, minerals, vitamins, etc. are transported by plasma proteins. So we'll discuss now one by one. Carbon dioxide is transported by the plasma proteins in the form of carbamino compound. So carbon dioxide is converted into the carbamino compound and they are transported by plasma proteins. Thyroxine is transported by an alpha globulin called thyroxine binding protein. So thyroxine binds with the alpha globulin and becomes thyroxine binding protein and it is transported then. Cortisol is transported by transcortin, which is a mucoprotein. Mucoprotein means having mucus within. So transcortin is a mucoprotein, so cortisol combines with it and it is transported. Then vitamin A, D and E are transported by the high and low density lipoproteins, that is HDL and LDL. Vitamin B12 is bound to transcobalamin for transport. So vitamin B12 is combined with transcobalamin and then it is transported through the body. Then bilirubin is associated with albumin and also with fractions of the alpha globulin. Then drugs of various types are transported after combining with the albumin. So various drugs combine with the albumin and then they are transported in the body. Then calcium of the plasma is partly bound to proteins for transport. So calcium binds to about 50% of the plasma protein for their transport. Then copper is bound to ceruloplasmin, that is alpha 2 globulin for transport. Then free hemoglobin in the vessels is bound by heptoglobulin and carried to reticuloendothelial system. So these all are the substances which are transported by plasma proteins and you have to remember all this. Then the seventh function is role as reserve proteins. Plasma proteins serve as reserve proteins and are utilized by the body tissues during conditions like fasting, inadequate protein intake, and excessive catabolism of the body proteins. So, plasma proteins reserve the minerals and nutrients, and so they serve as reserve proteins, and then when the body needs it, when the body is starving for these nutrients and minerals, then these reserve proteins are used by the body tissues in various conditions. And these various conditions are like fasting, inadequate protein intake, and excessive catabolism of the body protein. The eighth function is role in suspension stability of RBCs. Suspension stability refers to the property of RBCs by virtue of which they remain uniformly suspended in the blood. So, suspension means remain freely in the air or in the fluid space. So, suspension stability is a property of RBC due to which they remain uniformly suspended in the body, in the blood. Globulins and fibrinogen accelerate this property. Then the ninth function is fibrinolytic function. The enzymes of the fibrinolytic system digest the intravascular clot, that is thrombus, and thus save from the disastrous effects of thrombosis. So, this enzyme is present in the fibrinolytic system, fibrins like system, digest the intravascular clot. That is, the if there occurs the thrombosis, then these enzymes in the fibrinolytic system, they digest the intravascular clot, that is this thrombus, and thus save the person from disastrous effects of thrombosis. Then the 10th one is role in genetic information. Many plasma proteins exhibit polymorphism. Polymorphism is a Mendelian trait that exists in the population with differing prevalence. So these plasma proteins exhibit polymorphism. Then 11th and last one is role of nourishment of tissue cells. So the plasma proteins are utilized by the leukocytes that to produce the substances known as triphones or chiral, which are essential for the nourishment of tissue cells. So leukocytes, that is WBCs, use plasma proteins to produce the nutritioning substances that are triphones or chiral, and they are used for nourishment of tissue cells. Now we'll discuss about the synthesis of plasma proteins. So in synthesis, we'll discuss about two things: the site of synthesis, or where does the plasma proteins are synthesized, and the factors affecting the synthesis of plasma proteins. So we'll start with the site of synthesis. In embryo stage and in adult stage, both are different. So in embryo stage, the plasma proteins are synthesized by the mesenchymal cells. First the albumin is produced and then the other proteins are synthesized. So out of the three protein, first of all albumin is synthesized, and they are synthesized by the mesenchymal cells in embryo. In adults, the plasma proteins are synthesized as described below. The albumin and fibrinogen are synthesized mostly by the reticuloendothelial cells of the liver. The alpha and beta globulins are synthesized by the liver, spleen, and bone marrow. And gamma globulins are synthesized by the B lymphocytes. So, as we have discussed, albumin, globulin, and fibrinogen. Globulin have three types: alpha, beta, and gamma. The albumin and fibrinogen both are synthesized by the reticuloendothelial cells of the liver, and alpha and beta globulins are synthesized in the liver, spleen, and bone marrow, and gamma globulins are synthesized by B lymphocytes. So in embryo, the plasma proteins, the whole as a group are synthesized by mesenchymal cells, while in the adults they are synthesized by different sites, that is in albumin and fibrinogen by reticuloendothelial cells of the liver, alpha and beta globulins by liver, spleen, and bone marrow, and gamma globulins by the B lymphocytes. Now we'll discuss the factors affecting the synthesis of plasma proteins. So first one is dietary proteins. Dietary proteins play the most essential role in the synthesis of plasma proteins. So dietary proteins are the most important in it. The relation of plasma proteins to diet was studied in the plasma protein depleted dogs, first of all by Whipple by an experimental procedure called plasmapheresis. So this experiment was done in a dog, plasma protein depleted dogs, means they don't have plasma proteins. Depleted means depletion. We say depletion means reduced plasma proteins. So the dogs which have reduced plasma proteins, in them the experiment was done, and first of all it was done by Whipple, a scientist named Whipple. So we'll learn about what is this plasmapheresis. Plasmapheresis, in this experiment the dog is rendered hypoproteinemic by repeatedly withdrawing whole blood and injecting back the cellular elements of the blood suspended in the ringer lock solution. So in then this process is repeated daily till the level of plasma proteins fall to 4 gram per 100 ml. Thereafter, different standard diets are given and their effects on protein synthesis are studied. So, what is the procedure of the experiment? It is like that the dog which is hypoproteinemic, they are made hypoproteinemic by repeatedly withdrawing the blood. So the whole blood in the body of the dog, it is withdrawn, and injecting back the cellular elements of the blood, and only the cellular elements are injected back in the body of the dog. uh, this process is repeated daily, and this process is repeated daily till the level of plasma proteins falls to 4 gram per 100 ml. So only a very few amount of plasma protein is to be injected, that is its level becomes 4 gram per 100 ml. Thereafter different standard diets are given and their effects on protein synthesis are studied. So well nutrition rich diet is given and its effect on the protein synthesis are being studied. So what are the conclusions of these experiments? So we'll see the conclusion. Dietary proteins are essential for the synthesis of plasma proteins. Proteins we take in our regular diet are essential for the synthesis of plasma proteins. The essential amino acids must be present in the diet for satisfactory synthesis of plasma proteins. So, amino acids should be present in the diet for synthesis of plasma proteins. Then, dietary proteins of animal origin favor albumin synthesis, while the dietary proteins of plant origin favor globulin synthesis. So, we have concluded that dietary proteins are essential. Then, essential amino acids should be present in the diet for the synthesis of plasma proteins, and albumin is synthesized by dietary proteins of animal origin, and the globulin is synthesized by the dietary proteins of plant origin. We have to remember all these 4 points, they are important in this process of plasmapheresis. Now, the second one is the other factors. Other many factors are responsible for it. So, other factors which affect plasma protein synthesis in the body are the following. There are many, like presence of infection in the body reduces plasma protein synthesis. So infection present in the body, they reduce the amount of plasma protein synthesis. Then exposure to some antigen stimulates formation of antibodies. So antibodies are stimulated because we have to fight against this antigen, because they are harmful to our body. So exposure to some antigen stimulates the process of formation of antibodies. Then inflammatory conditions promote the synthesis of a number of proteins. So inflammatory conditions accelerate the synthesis of proteins. Then interleukin 1, a material released by the activated macrophages in the body, stimulates the synthesis of many acute phase proteins in the liver. So interleukin 1, it stimulates synthesis of proteins in the liver, acute phase proteins in the liver. And this interleukin 1 is released by activated macrophages in the body. Then prostaglandins are also reported to increase the synthesis of acute phase proteins, possibly through stimulation of macrophage release of interleukin 1. So prostaglandins also increase the synthesis of acute phase proteins by stimulating the macrophage release of one interleukin 1. So these were the factors affecting the synthesis of plasma proteins. In case of any doubts, you can ask in the comment section, and please like the videos and share it with your friends, and don't forget to subscribe the channel. Thank you.